आइए हम सुने विज्ञान की अनकही कहानियाँ। आइए हम सुने विज्ञान की अनसुनी कहानियाँ। एक नई दुनिया की आवाज़ आपकी आवाज़, विज्ञान की आवाज़, की आवाज़।
We bring you all a podcast series on STEM popularization, that is, science, technology, engineering, mathematics, and medicine, supported and catalyzed by the National Council for Science and Technology Communication, the Government of India. Program is produced by Media for Community Foundation.
Welcome back to the podcast from the Indo French Center for the Promotion of Advanced Research, also known as CIFIPRA today. We are dedicated to showcasing the groundbreaking collaborations between Indian and French scientists that are expanding the frontiers of knowledge. Now, let's understand more from a scientist.
Myself, Professor Sathees Raghavan. I'm a professor at Department of Biochemistry, Indian Institute of Science, Bangalore. I have joined here sometime in 2006 as an assistant professor in the Biochemistry Department, and then later I became associate professor and more recently professor. I'm basically from Kerala. I mean, initial education is from Kerala up to my master's. And then I went for my PhD in Banaras Hindu University, Department of Zoology, Varanasi. And once I completed my PhD in the area of DNA, damage, repair connected to germ cells, I moved to United States for my postdoctoral research. I joined at USC, University of Southern California in Los Angeles. And there my research was more focused on genetic abnormalities related to cancer, like the mechanisms, how some of those abnormalities are generated in normal people, and later it causes or those cells get transformed into cancer and things like that. And sometime in 2006, as I said before, moved to India back and then Indian Institute of Science. And currently my lab is focused on different aspects of DNA damage repair and their connection to cancer. For example, we work quite a bit on DNA double-strand break repair, which has also some extent connection with the CIFIPRA and this project which I'll be talking later. And we work particularly on a DNA double-strand break repair pathway called non-homologous enjoining, which takes care of most of the DNA breaks inside human cells. Mostly. Another aspect we work on is related to VDJ recombination, which is one of the processes by which antibody diversity is generated. So its connection to again cancer and how sometimes the VDJ recombination rearrangement cause or lead to oncogenesis in some of the aspects we study. Besides, we also work on, or try to develop some of the small molecule inhibitors that can be used for treating cancer. So these are like three major focus. Now, coming to this particular project, CIFIPRA project, this was focused on identifying novel non-homologous enjoining inhibitors which can be used for treating cancer. Now, my French collaborator in this case was Dr. Professor Jean-Baptiste Chabner. He is from Paris, Saclay. So the beginning of the project was essentially because of another discovery from my lab, in which we described the first NHJ inhibitor. NHJ is non-homologous enjoining. NHJ inhibitor from the lab. It was published in Cell, one of the topmost journals in the world. And then, so that's where JD contacted me, and then he has like real good expertise in working with non-homologous enjoining, and he's a crystallographer. He has identified several important factors related to NHEJ. So that's where the origin. We discussed, and then we thought, why don't we try to come up with a new small molecule inhibitor better than what we described in our original discovery, because there were certain limitations with respect to that step. So that's it. So that's where our discussion initiated, and then which culminated in this particular CFIPRA project, wherein we want to make new generation NHJ inhibitors and then using it for cancer therapy, which I'll explain shortly. Yeah, so as I mentioned, this project essentially deals with the DNA repair inhibitors, and when we started this, our interest was to look for small molecule inhibitors with better efficacy, and that can be used against treatment of cancer, wherever NHJ is non-homologous enjoining is upregulated. That was one of the objectives, and then another major objective was to look after radio-resistant and chemo-resistant cancers, because one of the major reasons for radio-resistant and chemo-resistance is upregulated DNA repair pathways. That's what people have reported, which essentially means that some of the factors or proteins responsible for these DNA repair process may get upregulated. And one of the enzymes which we were interested was ligase 4. So now the ligase 4 is an enzyme that is responsible for final joining of broken DNA during NHJ pathway. Okay. So we targeted this enzyme, especially the DNA binding domain of the enzyme, and then tried to develop or come up with the small molecule inhibitors in our original study. As part of CFIPRA project, so we tried to improve the efficacy of these molecules, because that was one of the real hurdles to taking forward this molecule to the clinics. Because if you don't have a small molecule inhibitor with efficacy at nanomolar level, or good efficacy in animal models, normally no companies will be interested, and it's almost impossible to take forward. So that's where myself and Professor JB had this thought, wherein we initiated this project and of course proposed, and then it got cleared later, and then we initiated this work sometime, if I remember, 2014 or 15, something like that. So some of the important findings from the projects include, as I said, first was identifying different forms of ACR7. So now the ACR7 is the first, the real NHJ inhibitor to be discovered. But as I said, there were certain limitations. So, and also we found that ACR7 actually can be present in multiple forms. It can get autocyclized, it can get oxidized into different forms. Our lab and JB's lab combinedly characterized these forms, and then later we went on to show that these molecules are specific to NHJ, and then it works in with good efficacy. And then second objective of this project was further improving the existing molecules. So that's where we have now come up with more recent, molecules with better efficacy, which are called now ACR 116 and 132. So these are the two molecules. Of course, to get this molecule, we synthesized many molecules. So these are all now published. Recently, one of the journals published this. And so what we found is the new generation molecules are approximately 100-fold better than the original molecules. So that's a big improvement. Earlier it used to work in micromolar level, 40, 50 or even 100 micromolar levels, but now we have this new molecule working at nanomolar level, which is another very important advancement in terms of finding. Then we tested these molecules in animal models with different tumors, wherein NHJ is upregulated. And then again, we could find significant improvement in terms of their efficacy with respect to tumor regression. So the bottom line, this project has yielded something. I would say still it's a starting point, but something really significant and which can be taken forward. Now, another aspect, or I would say maybe third objective, which was initiated as part of this project was looking at the toxicological effect of this molecule. So as you may know, whenever we have these kind of discoveries in the lab, like identifying a chemical like this, see, efficacy is one of the aspects, but then if the toxicity is not taken care, this molecule can be never taken forward. See, that's the truth, right? So that is what was our next approach, and then we could not, of course, conduct all the studies because that was not the mandate of the project described in this. But at least we conducted several initial preliminary toxicological studies, and we have gone up to now 28 days toxicity. And what we found is it does not cause much harm as far as normal cells are concerned. Now, one of the relevant questions always asked to us was, NH is also involved in one of this process where immune diversity is generated, which is called VDJ recombination. Since that is also, this particular process is also, DNA repair pathway is also involved in this process, we try to look at what will be the side effect in terms of immune system. So what we found is something very promising. At least the kind of concentration level we test in the animals or cell lines, we do not find much problem in terms of T cells and B cell proliferation. But then again, of course, we will be investigating these things in much more details. Yeah, see, if you look at this example, whatever I just talked about our research itself, see what you see is, see the kind of, the magnitude of the problem what we are trying to address, trying to find out, trying to have some basic discovery, then trying to incorporate the translational relevance, then have a vision to take it forward probably to the next level. See, these kind of projects, conducting in a normal scenario, getting funding, it would have been very difficult for us, right? And then now the other aspect, which probably I did not talk much about my collaborator, see his expertise in terms of doing a lot of these experiments associated with this, like he's a crystallographer, he's a biophysicist. So this complementarity helped a lot for evolving this project to the current scenario. That would not have been possible, like say if an Indo French collaboration was not possible, right? So not only the kind of funding was supported and the expertise shared between these two countries, and then the other aspect, see, we have also a lot of collaboration at the level of students, which I did not even mention, right? If you look at our publication, you will see there are French students involved, Indian students involved. Like say, for example, my student, PhD student, visited my collaborator's lab. And of course, as it was a mandate for us to visit, but then, even students visited, they spent quite a lot of time and then learned new things. So that way, with respect to our project or with respect to any other project, these kind of a program is, in my opinion, it's like really, really helpful to grow the research and also the relation between two countries in terms of collaboration at the level of research. I think such kind of programs, including whether it's Indo French or even with India and other countries, are very, very important. Because it gives a lot of opportunities, not only for Indian counterpart, even for the other people as well. So therefore, it helped a lot, particularly for our program. But I'm sure there would have been many other labs got benefited because certain things which we cannot do alone, maybe certain expertise which is lacking with us. It can be shared, and then I think that is the way forward. I think that should be our focus in coming years, wherein we should have such many more collaborations with different countries, and then that will give us give the opportunity for our people, Indian people, to grow as well as people.
This episode is brought to you with the content support from Cefipra and the technical support from the Sparrows team. We extend our heartfelt thanks to the Director of CIFIPRA, Professor Nitin Seth and Dr. Payal for their continuous guidance and support. Stay tuned for more episodes and thank you for joining.
आइए हम सुने विज्ञान की अनकही कहानियाँ। आइए हम सुने विज्ञान की अनसुनी